So, welcome everybody. I can see myself live. So, let's start today's session. So, today's session is on a very important topic called as triacylglycerols. Okay, so it's basically from lipid metabolism where we are going to study. So, for anyone who is attending my session for the first time, I'm Dr. Abhishek, your mentor for biochemistry at Unacademy. I'm also the author of Biochemistry Basics flashcard, which is available on Amazon. And you can follow me at Unacademy to get a notification of all my live classes. Okay, if you are new to Unacademy channel, make sure that you subscribe to our channel and hit the bell icon, because not only me but more than 50 different educators come live every month on Unacademy. Whenever we have time, we take live classes on YouTube, uh, Unacademy channel. And very high topics, latest topics, and that are covered with this live classes. And if you have attended any one of my previous session, make sure that you hit the thumbs up button, because it will get it saved in your liked history. So these are some of the sessions which I'm taking on Unacademy platform. Again, these are all free sessions. These are all special classes, we call it. And as I told you, that whenever we get time, we try to take these special classes. So two sessions I've already taken, one on galactose metabolism, one on hexose monophosphate shunt. The next session is on fatty acid synthesis. That is going to be happening, tomorrow. And then fed and fast cycle, and then again there will be a session on triacylglycerols on the main Unacademy platform. Before I start today's sessions, let me talk about our new batch which we have just started. MBBS first prof batch. Now there were so many students who were first year students who were joining an academy, and they were consistently requesting for first prof batch, because they were not immediately going to take the NEET PG or the PG entrance examination, but they required a lot of in terms of how to write their answers, how to get good grades in their first prof examination, and how to learn their practical biochemistry, which is very important for the first prof. So we have this MBBS for first prof batch, which we have just started 2 days back on 12th. So the idea is basically to give the first year students a complete coverage in terms of university of prof exams, and keeping the next pattern in mind. So it will be a very in-depth class, and the classes will happen on alternate days. Like yesterday I had a class, today is a break, again tomorrow I'll be having, and Saturday Sunday generally will be off, so that they can easily attend this batch along with their college. And the timing of the batches will be 6 to 8 and 8:30 to 10:30. And again, I'm taking biochemistry in that in just 36 sessions. The entire biochemistry, the practical part, everything will be covered. Dr. Pradeep will be taking anatomy. He's also one of the best faculty in anatomy. And Dr. Somin Manna, one of the top faculties in physiology, is going to teach you physiology in this batch. So these are the three first year, yeah, and we have started in May. By November, your entire syllabus will be completed along with your college year. So these are the college highlights. So in case if you have any doubt, you can go and check out an academy app, and there you will go. So having said that, let's start with today's topic, triacylglycerol. So first of all, let's understand a very important concept. So let's look at this question. So can anyone answer this question? I'm going to show you a few questions, and then we'll look at the answers of these questions, and then we'll learn the topic. A 10-month-old boy with a seizure disorder is brought to a physician by his mother because of a 2 days history of vomiting and lethargy. Lab study shows decreased serum glucose concentration. Further testing confirms an enzyme in lipid metabolism that is found in liver. So there is an enzyme which is found in liver but not in adipose tissue. Which of the following enzyme is most likely to be deficient in this patient? So these are the new pattern questions which we are going to get. So the options are glycerol kinase, acetyl CoA carboxylase, glycerol 3-phosphate dehydrogenase, and acyl CoA dehydrogenase. So anyone wants to take a guess at this question? So even though the clinical history is given, but you can see in this question there is no relevance of the clinical history and the question which is asked. So that's why I say that whenever you are attempting a clinical question, make sure you read the options first, the last two lines of the question first, and then you read the entire question. Anyone wants to answer this question? Anyone? So We are going to, so this question we are going to answer in the session, that which is the enzyme which is present in the liver but adipose tissue lacks those enzymes. Let's look at this question which was asked in Zipmer again on the same topic. I'm not going to give you the answers. I'm going to teach you the concept and then you will automatically know the answers. Hormone sensitive lipase does not remove carbon from which, from triacylglycerol. Which carbon of triacylglycerol? So first carbon, second carbon, third carbon, or it removes from all the three carbons. So what is a hormone sensitive lipase, and what carbon does it not remove, or does it remove from all the carbon? Any question, any guesses on this? Anyone wants to answer this question? Hormone sensitive lipase does not remove carbon from triacylglycerol. Which carbon of triacylglycerol? Anyone? So again, I'll answer this question when I'm going to teach you, right? This is the third question, just as a preview. Regarding synthesis of triacylglycerol in adipose tissue, all of the following are true except. So which of the following is a true false statement you have to, because it is asking for a true except. Okay. So synthesis happens from dihydroxyacetone phosphate. Enzyme glycerol kinase plays an important role. Enzyme glycerol 3-phosphate dehydrogenase plays an important role. And phosphatidate is hydrolyzed. So which of these is a false statement? Which of these is actually a false statement? So these three questions I have shown you. Okay, and now we'll go to the theory. Okay, so let's start by understanding triacylglycerol. So what is triacylglycerol? So basically, to understand triacylglycerol, you have to understand an important concept, that fatty acids are polar molecules. If you see, you know, if you see, this is a fatty acid. Okay, and one end of the fatty acid has a very active functional group, see this CWH. So whenever metabolism has to happen for the fatty acid, this is good. But when this fatty has to be stored or transported, this CWH is a very reactive group. Okay, so it has to be converted into a non-reactive group so that it can be stored. And how is this achieved? We combine, this is an acid, so it can easily combine with a base or an alcohol. So we have this glycerol molecule. Okay, and it has 3 OH. Okay, and there will be 3 fatty acids which will have 3 CWH. So what kind of reaction we are looking at? We are looking at a CWH plus OH. Okay, this OH and H will be removed as water, and what we will have is a C double O bond, which is also called as the ester bond. Is this clear? Ester bond. So what will happen? This glycerol molecule will react with 3 fatty acids. Now remember, these 3 fatty acids can be same or different. Okay, and what do they form is something called as a triacylglycerol. So this will form into C, 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 and here we have got seen a ester bond and then the fatty acid. So let's just keep it as fatty acid only. So this is your triacylglycerol. Is the concept of triacyl clear to all of you? What is a triacylglycerol? So basically a glycerol molecule with 3 fatty acids attached to it with a ester bond, with a ester bond. And what is the significance of this triacylglycerol? This is basically the storage form or the transport form of fatty acids. Is this clear to all of you? What is the significance of this triacylglycerol? It is the storage form or the transport form of fatty acid, because as I told you, fatty acids are, at one end it will have a very reactive compound. So yes, it can go into all the metabolic pathways, but for transportation and store, this is not a good way. Is this clear? First of all, what is triacylglycerol? So what happens? This fatty acid, this functional acid group reacts with a OH from this, okay, from glycerol molecule. If you see, this is a glycerol molecule. So 3 OH will react with 3 fatty acid, and this is how your triacylglycerol is formed. Okay, overall. Having said that, what kind of lipids are triacylglycerol? Are they simple lipid? Are they compound lipid? Are they derived lipid? Or are they miscellaneous lipid? Can anyone answer them? This is the Bloor classification of lipid. So what kind of lipid is your triacylglycerol? Again, this was a very important question which has been asked. So remember, it belongs to the simple lipid. Why? Because if you see, what are simple lipid? By definition of Bloor classification, simple lipids are esters of fatty acid. Simple lipids are esters of fatty acid with alcohol. So exactly that is what we have seen in triacylglycerol. So there is a ester bond formation of the alcohol and the fatty acid. Okay. So by this definition, triacylglycerol is basically a simple lipid. Okay. Whereas contrary to your belief, you know whereas both glycerol and fatty acids, individually are classified as derived lipid. Is this clear? So derived lipid is something which can be derived from simple or compound lipids. So these are derived, classified as derived lipid. Again, be very careful when you are answering this question. Okay. So basically, this triacyl glycerol can be in solid or liquid form. If they are in solid form, they are called as fat. If they are in liquid form, they are called as oils. Okay. And compound lipids, we know that these are basically simple lipid with a prosthetic group. Anything attached to the simple lipid. For example, if you have phosphate attached, it will become phospholipid. If they have uh, you know carbohydrate molecule attached, they will become glycolipid, or proteins attached, they will become lipoproteins, or sulfur attached, they will become sulfolipids. So very important that all these compound lipids are basically going to be derived from a triacyl glycerol. So when you're going to see phospholipid, they will have the basic structure of triacyl glycerol, and in addition there will be a phosphate group. Okay. So having understood the classification, let's quickly look at the digestion of lipid, because one again very important point you have to remember is triacyl glycerol composed of 97% of your dietary intake. Triacyl glycerol is made up of 97% of your dietary intake. Okay, so 97% of your diet is composed of triacyl glycerol. So to digest triacyl glycerol, we have got these enzymes: lingual or gastric lipase, then you have got pancreatic lipase, then you have got pancreatic esterase, and then you have got phospholipases to remove phospholipid. Okay, so this is the all the enzymes which is there for digestion. Now what happens, each of these, so what happens is, first lingual and gastric lipase will remove the third ester bond. So we have told you there are three ester bonds in your you know in your glycerol molecule. There are three ester bonds. This first bond is broken by, third bond is broken by lingual or gastric lipase. Okay, and almost 30% of the digestion happens in stomach. Then this entire diacyl glycerol comes to the small intestine, where pancreas lipase will further break the first bond. Okay, so pancreatic lipase breaks the first bond, and now you are left with a monoacyl glycerol, which means only on the second carbon you have got fatty acid, because these two fatty acids have been removed, and these are removed by your pancreatic esterases. So is it clear? Is it clear? So what happens, first the lingual or the gastric lipase breaks the first bond, then pancreatic lipase breaks the second, third bond, and then eventually one acyl molecule is left, and that will be broken by pancreatic esterase. Okay, and phospholipases will bring the phospholipids. Okay, a small side point on bile salts. So what are bile salts? What is the role of entire bile salts in absorption of lipid or metabolism of triacyl glycerol? So they act as biological detergents. Okay, so these bile salts act as a biological detergent and help in emulsification of products like lipid digestion, you know and the minimal bile salt concentration for formation for this emulsification is called a critical micellar concentration, which is around 5 ml. Okay, and all the fat soluble vitamins are transported in micelles. Okay, so again, digestion of we have seen that digestion is very very important. Now very important, what happens is, we have seen that three all the three fatty acids of your glycerol is broken as it is digested. Okay, so by the time it is reaching the small intestine, it is basically back into glycerol. Okay, and all the free fatty acids are there. Now, all the free fatty acid, whatever is short chain or medium chain, whatever is short chain and medium chain, will directly get absorbed, okay, into the portal circulation. But all the long chain and very long chain fatty acids, what will happen to them? They cannot be directly absorbed. So what happens? These again combine with this, okay, and now again triacyl glycerol is formed. So when you ate your food, you got maximum as triacyl glycerol, but your enzymes broke all those triacyl glycerol into glycerol and free fatty acid. And what happened? That free fatty acid, whatever was small or medium, was absorbed, but whatever long and very long was again combined with the glycerol to become triacyl glycerol. So what is the difference basically? What is the difference? So if you see the major difference is, when you eat food, the triacyl glycerol in the food was composed of all kind of chains. So it had short chain fatty acid, it had long chain fatty acid, it had medium chain fatty acid, very long chain fatty acid. But the triacyl glycerol, which is formed in the small intestine so that it can be loaded into chylomicron, will have only very long chain or long chain fatty acid. They will not have short chain or medium chain fatty acid. Is this clear? Is this clear to all of you? So this is very, very important for you to understand. And this, we know that this triacylglycerol will loaded into chylomicron and this will be absorbed. So this is about absorption of triacylglycerol. Now let's, so this was about exogenous triglyceride. Exogenous means whatever you are taking in terms of food. Now let's look at endogenous triglyceride. Endogenous means, you know, endogenous means whatever is formed. We're gonna, our liver also forms a lot of fatty acid. So how that fatty acid is actually converted into triacylglycerol, that is the most important here. So predominantly where does the triacylglycerol is formed? Okay, it is formed in your liver, it is formed in your adipose tissue, and it is formed in your intestinal mucosa, and majority in endoplasmic reticulum. So there are three steps to this triacylglycerol synthesis, and remember here we are talking about endogenous triacylglycerol synthesis. Earlier whatever we have talked was exogenous, which means whatever we have taken into the, from the food, how that triacylglycerol is being metabolized. Now this set of the, things which are going to is about endogenous, which means whatever fatty acid we are going to eat. So the three steps are: first, activation of fatty acid. So whenever fatty acid is formed, it will be reacting with CoA to be activated. So fatty acid CoA will be formed. So that is the first step, activation. Then activation of glycerol. So glycerol molecule also will be activated. It will be added on with a phosphate group to form glycerol, phosphate. And here the enzyme is kinase, glycerol kinase. So this is activation of glycerol kinase. And then on the glycerol molecule, one by one, each of these fatty acids will be added. So let's look at each one of these steps. So activation of fatty acid generally happens with acyl-CoA synthetase, also called as thiokinase, and fatty acid is converted into CoA derivative. Okay, glycerol kinase is the enzyme for activation of glycerol, and this here glycerol is converted to glycerol 3-phosphate. So if you remember the structure of glycerol, this was glycerol structure. So on the third carbon you have a phosphate which is attached. Okay, so this is your glycerol phosphate, and this reaction is converted by glycerol 3 glycerol kinase, and then one by one fatty acid is being added. So we will look at the steps. Now remember, this is a very important, and most of the questions I have shown you at the start of the session was on this topic only. Glycerol, with the help of glycerol kinase, is formed into glycerol 3-phosphate. So this step is called as the activation of glycerol. Now remember, it happens in liver. Okay, it happens in adipose tissue, muscle, and endoplasmic reticulum. The problem is it happens not in all adipose tissue, it happens only in the brown fatty tissue. Normal adipose tissue, this reaction will not happen. Okay, right? So this, and so because normal adipose tissue lacks this glycerol kinase, this is a very, very important, and this question has been asked. Glycerol kinase is not present in the white adipose tissue. Okay, so remember, whenever they will ask you what is the enzyme which is present in liver and not present in the adipose tissue, the correct answer will be glycerol kinase. So what happens in this white adipose tissue? How do they form this glycerol 3-phosphate? Basically they convert dihydroxyacetone phosphate into glycerol 3-phosphate. Okay, they convert dihydroxyacetone phosphate into glycerol 3-phosphate in the adipose tissue. So now let's happen. So what happens? Glycerol, with the help of glycerol kinase, is converted into glycerol 3-phosphate, and in adipose tissue it will be dihydroxyacetone phosphate, which will be converted, dehydrogenated into glycerol 3-phosphate. Then acyl transferase will add the first fatty acid. So what happens? Suppose this is your glycerol 3-phosphate. Here OH is there. Here OH is there. Now the first acyl transferase, what it will do is it will react and add a fatty acid here. Okay, so this structure is called as a lysophilic acid. Is it clear? This structure is called as a lysophilic phosphatidic acid. So in lysophosphatidic acid, first carbon has a fatty acid, second carbon has an OH, and third carbon has a phosphate group attached. Then what happens? Another acyl transferase will add another phosphate group. So already, here already fatty acid was added, here phosphate was added. Now another fatty acid will be added here. So this structure, okay, the second structure, this is called as the phosphatidic acid. Is this clear? So phosphatidic acid again is a very important, because all the phosphates will be formed here. Okay, so again what was happening is, let me just tell you till now what has happened. So glycerol, this was your glycerol molecule, this was your glycerol molecule, and here instead of this OH, first phosphate was added. So glycerol kinase into glycerol 3-phosphate. Then what happened? Another first fatty acid was added here. Okay, then the second fatty acid was added here. So what we have is something called as a phosphatidic acid, fatty acid 1 here, fatty acid 2 here, and here you have got phosphate. Phos Now what happens, with the help of phosphatase, that phosphate is removed. So here, this was the structure till now, fatty acid, and here phosphate was here. Now this phosphatase, what it will do, it will remove the phosphate. So this phosphate is now removed. And now what we are having is something called as a diacyl glycerol, because you can see two fatty acids here, and here glycerol backbone is there. Diacyl because two fatty acid is there. And then the last, again with the help of acyl transferase, again this, the third carbon from which just the phosphate we have removed, again fatty acid will be added, and now you have got your triacyl glycerol. Is this clear? So it's a multi-step process. First the glycerol is activated, first fatty acid is added, second fatty acid is added, then the phosphate is removed, and the third fatty acid is added. Is it clear to everybody? Is it clear to everybody how synthesis of triacyl glycerol happens? Okay, now let's see how they are broken. Okay, how the breakdown. So this was all about synthesis of triacyl glycerol. Now let's look at how these triacyl glycerols are broken, also called as lipolysis. So this triacyl glycerols, to be broken, so what it will be breaking into, so remember this is your structure of triacyl glycerol. Okay, so what will it break into? With this help of this class of enzyme called as lipase, this triacyl glycerol will break into your glycerol molecule, okay, and your three free fatty acid. Whatever these three free fatty acid was there, that three fatty acid will be, okay. And this hydrolysis, lipolysis generally happens in the adipose tissue, okay. Remember this free fatty acid, when they are released into the plasma, they bind with serum albumin, and how that is how they are, yeah. Free fatty acid can be taken up by liver, cardiac tissue, kidney, muscle, lung, testis, and adipose. But brain, because this albumin and free fatty acid, cannot combine, cannot cross the blood-brain barrier. So brain will not take up this free fatty acid. Okay. Another very important point: glycerol kinase is present in liver, kidney, intestine, and brown adipose tissue. Okay. So remember, brown adipose tissue has glycerol kinase, whereas white adipose tissue lack glycerol kinase. And this is the question which they have very frequently asked. Is this clear? Brown adipose tissue. So how does white adipose tissue form this glycerol 3 phosphate? They will convert dihydroxyacetone phosphate with the help of dehydrogenase enzyme into glycerol 3 phosphate. Okay, so this is the mechanism. Okay, there's a very important, so these lipases can be very of various kind. For example, hormone sensitive lipase we are going to study, lipoprotein lipase will be there, acid lipase will be there. So there are so many different types of lipases which are there. Okay, so especially you have to learn about is hormone sensitive lipase. This hormone sensitive lipase is activated by glucagon. Okay, this is activated by glucagon and inhibited by insulin. Remember this, very very important. Hormone sensitive lipase are what are controlled by the hormones, the glucagon and the insulin. Okay, so hormone sensitive lipase, the characteristic feature is it will remove the fatty acid from the first and the third carbon, but it can never remove from the second carbon. So suppose this is your glycerol molecule. Okay, when hormone sensitive lipase acts on it, it will convert it into this. Okay, two fatty acids will be removed, but the second carbon fatty acid will always be attached. So this again was the question which was asked. Hormone sensitive lipase cannot remove free fatty acid from the second carbon. Okay, so what enzyme removes it? Is removed by 2-monoacyl glycerol lipase. Okay, again hormone sensitive lipase is activated by glucagon and inactivated by insulin. Okay, and in diabetes you have a hormone sensitive lipase which is activated. So this was all about degradation of it. Okay, so let's quickly look at all those three questions which you have seen. Okay, so this is the first question. So now can you all of you answer, now can all of you answer this question? A 10-month-old boy with a seizure disorder is brought to physician by his mother because of a 2-day history of vomiting and lethargy. Labs study shows decreased serum glucose concentration. Further testing confirms a deficiency in an enzyme involved in lipid metabolism that is found in liver but not in adipose tissue. Which of the following enzyme is most likely deficient in this patient? Anyone wants to answer this question? So what is the function of acetyl-CoA carboxylase for fatty acid synthesis? Okay, so this is present both in liver as well as in adipose tissue. But as I told you, glycerol kinase is only present in the liver but never in free fatty tissue. Okay, especially the white adipose tissue, it is not present. Glycerol 3 phosphate dehydrogenase, again it is present both in liver as well as in free fatty. Acyl CoA dehydrogenase for beta oxidation, again it is present in both. So the main enzyme which is present in liver but not in adipose tissue is glycerol kinase. Is this clear? Let's answer this question, all of you. Hormone sensitive lipase does not remove which carbon from triacylglycerol? Hormone sensitive lipase does not remove from which does not remove. So I have told you already, triacylglycerol, three fatty acids are attached. So which fatty acid will not be removed by hormone sensitive lipase? So remember, this is removed, this is removed. Only this fatty acid is not removed. This is not removed. So once hormone sensitive lipase has acted on it, what you will have is 2 monoacyl glycerol. So here free fatty acid will be attached. Okay. So this fatty acid first and third position will be removed by hormone sensitive lipase. Priyanka, is this clear? The second carbon free fatty acid will not be removed. Is it clear? Excellent. Okay, let's look at the third question regarding triacylglycerol in adipose tissue, synthesis of triacylglycerol. All of the following are true except all of the following. Let's answer this question regarding synthesis of triacylglycerol in adipose tissue. All of the following are true except synthesis from dihydroxyacetone phosphate. So we know that in adipose tissue, because glycerol kinase is not there. Okay, glycerol kinase is not there. So dihydroxyacetone phosphate with the help of dehydrogenase, will become first is will be converted to glycerol 3 phosphate. Okay, so this is how it will be formed. Okay, so this is correct, this is correct. We have seen that glycerol kinase is absent in adipose tissue, so this cannot be correct. And phosphatidylate is hydrolyzed, this is also correct. Okay, so the only incorrect option is B. Okay, enzyme glycerol kinase is plays an important role. No, enzyme glycerol kinase is absent in your white adipose tissue. So this brings us to the end of our session. So a little reminder about Plus subscription. You know that at Unacademy we try to give as much as free classes as possible, but you know that whenever we are available, then only we can give free classes. When you join Plus subscription, you will get daily classes. They are more structured and unlimited access you can get. You know that Unacademy now has Prep Ladder along with it, so you can get both the benefit of Unacademy and Prep Ladder in our iconic subscription. There's a limited time offer, like for 36 month only 58,000 and more. If you use this discount code Dr. Abhishek 10, you'll get a 10% off. Okay, so this is, yeah, and all, even if you take for 1 year, there will be a huge discount on that. If you are already going to appear for NEET PG this year, this packs makes a lot of sense. 3 month, if you buy, you get 1 month additional, yeah. So again, this is till the offer lasts. And no matter what exam you are preparing for, there's always a batch at Unacademy. For example, this is a batch for anyone who was supposed to attempt this year, but now, because of some reason, because the exam has been postponed, so they're not able to give. Okay, so this is just to make sure that they are actually practicing and learning, and not they don't lose track of their preparation. FMG batch, we have some excellent results in our FMG students. So FMG batch is there. First MBBS first prof BAP I already discussed. I am a part of that. Dr. Pradeep is there, Dr. Somenda is there. And then we have got target next batch. So for next year target, we have got an evening batch again. The idea is to complete it the syllabus by November, so that you can practice a lot of questions and are absolutely prepared. There is an INI CT batch, which is again a part of Unacademy. Very nice question bank of 34,000. Whatever questions I show you, most of them are from Unacademy question bank. So if you have an access to this bank, you can solve it on your own, so that you can practice well. And so this is all about it. And make sure that you have subscribed and hit the bell icon. And if you have watched till this place, make sure that you give this video a thumbs up. Okay, and thank you everybody. This is the discount code. Either you can use Dr. Abhishek -YT, or also if you want, if you can use Dr. Abhishek 10, because 10% discount it will give you, Dr. Abhishek 10. So thank you so much for watching this session. And tomorrow's session, again, let me just check what is tomorrow's session. Just update you on tomorrow's session. Okay, so tomorrow we have a very important session on fed and fast cycle. Fed and fast cycle. So I'll bring the questions. I'll discuss the concept. Many students, this is a very important concept from exam point of view, from biochemistry. Direct questions, clinical questions are made around that fast cycling. So thank you so much, everybody, and make sure that you give this video a thumbs up, you share it with your friends so that it reaches more and more students. Thank you all.